So scientists just did something pretty incredible. They've created a completely seamless, gap-free genetic blueprint of an animal that, believe it or not, is unbelievably important to our own health. We're talking about the domestic pig. And this new, perfect genome is changing, well, just about everything. Okay, let's just get right to it, because it's a fair question, right? Of all the animals on Earth, why pour all this time and energy into a pig? Well, as you're about to see, this goes way beyond the farm. This is really about unlocking secrets that could totally revolutionize human medicine.
And our story starts not with just any old pig, but with a very, very special one from China.
So, meet the Wuzhishan pig. It's well known for being really small, maybe half the size of a typical commercial pig. And that unique trait, that small size, makes it an amazing model for biomedical research. I mean, think about it. It's easier to handle, and the costs for food and housing are way lower, which is a huge deal for scientists studying really complex human diseases like diabetes.
But there was a major roadblock. The best genetic map we had for pigs, the instruction book for how to build a pig, was full of holes. The old reference, called Scrofa 11.1, had over 500 gaps. Imagine you're trying to read a critical instruction manual, but hundreds of pages are just gone. That's exactly what researchers were dealing with.
So to fix that, scientists turned to some pretty revolutionary technology to create the first ever truly complete genetic blueprint for the pig. And that breakthrough is called telomere-to-telomere, or TTT sequencing. Okay, think of a chromosome as a chapter in that instruction book. The old tech could only read like short sentences, and it had a really hard time piecing them together, especially in the tricky, repetitive parts. But TTT technology, it can read the entire chapter, from the very first word to the very last, from one end cap, the telomere, to the other. And that means no missing words, no jumbled sentences, just a perfect, seamless read. Now, just look at the difference here. It is staggering. The old genome had 506 gaps. The new one has 0. Zilch. Over 66 million base pairs, the actual letters of the genetic code, were just kind of floating around, unplaced. Now, they're all perfectly mapped. They even found almost 1,000 new genes. So this isn't just an update, it's a complete game changer. And to put a number on it, this just shows you the sheer magnitude of what they found. In total, the T2T genome unlocked over 194 million base pairs of previously unknown, unresolved genetic information. That is a massive amount of new biological territory to go out and explore. So, what was actually hiding in all of that new genetic data? What secrets did they find? Well, it was a treasure trove, basically. Scientists found 1,189 new genes that code for proteins. For the first time ever, they saw the complete, unbroken structure of all 20 pig chromosomes. And that gave them their first glimpse into some of the most mysterious and complicated parts of the genome, like the centromeres and the entire male Y chromosome. So let's quickly talk about that. A centromere is that pinched-in part of a chromosome, kind of like a central anchor point. It is absolutely essential for making sure cells divide the right way, but its DNA sequence is insanely repetitive, like reading a paragraph that just says the over and over. And that repetition has made centromeres this huge black box in genetics. Until now. And here, for the very first time, we can actually see them. This amazing imaging technique, it's called FISH, uses these fluorescent probes that are like tiny little beacons that stick to specific DNA sequences. It's like flipping on the lights in a part of the genetic map that has always been completely dark. Researchers could finally see the exact location, the structure, making the invisible visible. So, with this new gap-free view, scientists started noticing things that were totally hidden in the genetic noise before. Like on some chromosomes, they found this really unusual and complex structural pattern, a kind of genetic signature that we've never been able to see this clearly. The specific name isn't what's important here, but what is important is that we're now seeing this intricate architecture of the genome in a way that was just impossible with the old fragmented maps. And another massive milestone was sequencing the entire male Y chromosome, from tip to tick. It's over 43 million base pairs long, and they found it has 111 genes. And get this, 42 of those genes, nearly 40% of them, had never been seen before. So this perfect blueprint, It isn't just about finding new puzzle pieces. It's about using them to understand the pig's entire story. Think of these 339,000 new genetic markers like brand new, super specific family names that we've just discovered. With these markers, scientists can basically act like genetic genealogists, tracing the relationships between different pig populations all over the world with incredible precision. It's letting them completely redraw the pig family tree, giving us a much, much clearer picture of how they migrated and were domesticated over thousands of years. Which brings us all the way back to our hero pig and its central mystery. After all of this, could scientists finally figure out the genetic secret to its tiny size? Yes, they could. And the answer is pretty fascinating. It all comes down to a single gene called GALNT13. Scientists found really strong evidence that this one gene has been specifically selected for in the Wuzhishan pig over generations. So what does it do? Well, to put it simply, it acts like a brake on the growth of cartilage cells. So here's how it works, broken down real simple. First, that GLN13 gene sends out a message to the cartilage cells. And that message is basically, hey, slow down, stop multiplying so fast. So with cartilage production slowed down, the pig's bones just don't grow as long. And the result is its signature small body. It's a really simple mechanism for such a defining trait. Okay, so we have a complete map, and we've solved a big mystery. But what does this all actually mean for us? The implications here they're massive, and they go way beyond the pig pen. For agriculture, breeders can now work with a level of precision we could only dream of before, selecting for healthier, more productive animals. For medicine, this is a total game changer. It means we can create way more accurate pig models for human diseases, which could dramatically speed up the hunt for new cures. And for evolutionary biologists, it's like finding a new Rosetta Stone for understanding the genetic history of all mammals, including us. At the end of the day, this work really elevates the pig beyond the barnyard. It solidifies its role as a critical partner in biomedical research. This perfect genetic blueprint is a shared resource that's going to help in the fight against some of our toughest diseases. It's a huge leap forward, and it leaves us with a really fascinating question to chew on. Now that we have the technology to create these perfect blueprints, what other corners of the animal kingdom should we explore? Which animal's genetic code should be the next one we unlock completely?